yes, I'm directly the associate editor scientist, but I don't think that's the only reason they picked me for this. It's also because I've been a bit of a futures fan and a futures student while the only courses in the country, if I can world, a master's in course in futures. So I think that's why they picked me for, because it's very probably the hardest of the sessions I've heard this afternoon. So bear with it, I think it's going to be great. It isn't going to be straightforward. How many of you, by the way, have got degrees in science or even A-levels? Good, great. I'll hold you to that and come back to you later to make sure you actually know that. So we won't pull any punches. Okay. Now, I think the point about the future and the future of healthcare and medicine is that by about 2050, there's going to be some 9 billion, probably more of us on the planet, of whom a large percentage, about 16%, that's 1.5 billion, just in case you can't be asked to do the maths, will be old. Now this creates pressure points, at least one important pressure point for healthcare and medicine. Obviously there are many, many more, obviously emerging economies, all sorts of things, but that's a big pressure point. So I think also rising expectations is going to be a big deal too, because we hear about genomic medicine, we hear about all sorts of stem cell discoveries. Everyone's expecting a lot from medicine. I mean, who knows if it can deliver or not, but that's kind of what we're going to try and do today. So I think the good news is that there is healthcare really is poised to deliver and to reinvent itself. I mean, how far and how fast is anybody's guess? But some of the emerging technologies like stem cell research, local genomic sequencing, nanotech, bionics, regenerative medicine, these are all things you've heard about. But I think in the next hour we're going to be looking at how real they are. We're going to meet some of the researchers who are closing in on some of this stuff, hearing about the impact that they're making right now and what they expect in the future. We're going to come at it from 4 angles, which I think is quite novel and quite nice. We're looking at attempts to transcend aging. We're going to look at why basic science, paradoxically, is needed more than ever. We're going to look at the combined power of nanotech and nature, and we're going to look at what stem cells might do right now. So, at the end, I very much hope there will be plenty of time for you to ask questions, so, just keep your eyes on our agendas and find out some questions. But first up, I'd like to welcome Aubrey de Grey. Now, Aubrey is quite a well-known figure in the transhumanist movement, which is all about transcending current limitations. He's also a biomedical gerontologist who's developed what he calls comprehensive plans for the medical elimination of age-rated ill health, which are strategies for engineered negligible senescence. Bit of a mouthful, eh? But that's why he's now called the Chief Science Officer of the SENS Research Foundation. That's the California-based foundation, which is really combating the aging process through this program, SENS. Hang on to that one, SENS. Anyway, his talk is called How Foreseeable Regenerative Medicine Will Bring Aging to an End. Big optimism, Aubrey. Let's have a hand for Aubrey.
Well, that was very much better. All right, Mother, thank you very much for being here. It's great to be here. I hate doing really short talks, so I've got far too many slides, and so I'm in danger of speaking far too quickly with that slide. Another reason why I am going to be short of time is because, as some of you may have heard, yesterday Google announced the formation of a company named Calico, which is some kind of subsidiary of Google. It's all rather unclear exactly how it's going to work, but which will be dedicated to the defeat of aging, which is quite nice. And I somehow feel that my advocacy role is beginning to become a little easier, which is rather pleasant after all these years. But anyway, so I'm going to give you a very brief overview of what SENS Research Foundation does and why it's important. I'm going to start by reminding you that aging is quite bad for us. It's very common for people to think of aging as in some way distinct from the diseases of old age, but that's nonsense. The only way a disease can be a disease of old age is by being part of aging, by being some aspect of the later stages of this process that goes on throughout life that is initially harmless because the body is set up to tolerate a certain amount of pre-symptomatic damage, but eventually the damage overwhelms the body, and these are the sorts of results we get. course, the fascinating thing about aging from a medical perspective is that we've been so unsuccessful in doing anything about it, by contrast to our very spectacular success over the past century or two in bringing most infectious diseases under control, using whether it's just sanitation or whether it's medicines or control of mosquitoes or whatever. Why is it that aging-related diseases have been so recalcitrant, so resistant to medical intervention? Well, it's simple, really. The problem is that, unlike infectious diseases, aging is a side effect of being alive in the first place. It's essentially something that you can't simply eliminate from the body, because to do so you would have to redesign the whole body, and we can't do that. So yeah, we can tolerate a certain amount of damage, but too much of it causes disease and disability. So we have to think about it that way, and this is just something that doesn't happen. The things I've highlighted in red here are things that are actually not terribly widely appreciated, or at least not terribly widely acted upon. The fact is, diseases of old age are universal. People don't think of them as universal. They think, well, some people get cancer, some people get heart disease, and so on. But everyone actually knows very well that all of these things are things that everybody will get it if they live long enough and don't get the others. Because practically they will kill you, right? So these are medical problems. Now, I've just said that we can't cure the diseases of old age in the same way that we can cure an infectious disease by eliminating it from the body. But that doesn't mean that we can't address it with medicine. We can address it with medicine if we just start from the fact of the matter, the fact that aging is this process of accumulating damage which eventually overwhelms the body. What this misguidedness means, in other words, what the irrationality, the idea of treating the diseases of old age as if they could be really cured in the narrow sense, what it means is an enormous amount of misallocation of funds. I've just given you American numbers here because I just happen to have them. But the same thing applies across the entire industrialized world. The National Institutes of Health, which is the body that is responsible for most medical research funding in the USA, spends about $30 billion every year, of which only 3% goes to aging, and that sounds bad enough, because of course the large majority of the medical industry, the budget of the medical industry, is spent on trying to address the diseases and disabilities of old age. But it's actually worse than that. Out of that 3%, only 1/6 or so is spent on actually trying to understand aging, as opposed to things like geriatric medicine or the social policies around maintaining the dignity of the elderly, things like that. And within that, $150 million or so, a tiny amount, probably less than what I said there, less than the $10 million I said there, is spent on translational gerontology. In other words, trying to develop medicines that will actually do something about aging. Now, the foundation that's been created around my work, SENS Research Foundation, is really small. We've only got a few years, and we only have a few major donors who agree with us going. And we are already spending on that same order of money, in the region of $5 million a year, which is about as much as the US government to try to actually do something about aging. That's pretty shocking. So, what's the problem? Well, as I said, there's this misconception about addressing the diseases and disabilities of old age, which is essentially to treat them like normal infectious diseases. And that's what I'm calling here the geriatric approach, attacking the pathologies of old age just as if they could be eliminated from the body. And it's easy to see that that's a non-starter if you think about it at all, because the damage which creates those diseases is continuing to accumulate in the body, so any attempt to alleviate those symptoms is going to become increasingly ineffective as the person gets older. For the past century or so, people have been gradually realizing this. Only a small number of people, unfortunately, but that's better than nothing. And that's what has given rise to what I'm calling here the gerontology approach to combating aging. The approach that says, let's try and clean up metabolism, let's try and learn from other species and from various experiments about the nature of aging in such a way that we might be able to slow down the rate at which this damage is created. But that's actually pretty much a non-starter as well. Not only are the pathologies of aging extremely numerous, so numerous that I decided to put them in small font. But also, the metabolism is extraordinarily complicated. This is a tiny little subset of what we know about how metabolism works, how the body actually functions. And even that, as you can see, is rather hairy, and it's pretty inconceivable that we would be able to somehow tweak this in order to, make it do something we want to do, namely the creation of damage at a substantially slower rate than it naturally does, without at the same time having unintended consequences. So sure enough, the gerontology approach had also utterly failed to deliver any substantial postponement of age-related ill health. It's not very surprising, especially if you take into account the fact that this is what we know about how metabolism works, which is completely dwarfed, as any biologist will tell you, by the astronomical amount that we don't know about how it works, even ignoring the stuff that we don't even know that we don't know. So, but there is another approach, which is what SENS is all about, which is what I'm calling a maintenance approach. Rather than trying to slow down the rate at which metabolism causes damage, or the rate at which damage causes pathology, we simply try to separate those two processes from each other. We uncouple them by periodically going in and repairing the damage before it reaches the level of abundance that is pathogenic, but after it's actually been created. So here we are not trying to interfere in metabolism, but we are being preemptive with regards to the diseases of our day, going like a geriatric approach. It's really exactly the same as what we do with simple man-made machines, where the methods that we use successfully to keep them going substantially longer than they were designed to. The reason why we have cars like this that are 100 years old, that were designed to last only 10 or 15 years, is because if we fall in love with them enough to do sufficiently comprehensive maintenance on them, then they just don't go downhill. Vintage cars simply don't have a mortality rate. The only reason that vintage cars ever go out of circulation is when their owners die and their owners' kids can't be bothered to keep them up anymore. So of course we don't do that with most cars, but we know that we can. And the only reason we can't do it with the human body is because the human body is more complicated and we haven't yet figured out all the details. But we're getting damn close. The big inspiration that allowed us to start on this whole avenue of research was that we could actually classify all of the various types of molecular and cellular damage which accumulate in the body throughout life and eventually cause disease into a very manageable number of categories. There's these 7 major categories that I listed here. And this is, it's a courageous statement that this would be an exhaustive classification, but I think it's safe, partly because it's been the same classification for about 30 years now, and secondly because I've been actually giving this classification explicitly for more than 10 years, going out there and challenging people to try to add to it, and nobody's done so. Now, the relationship between aging and age-related disease is quite easy to see if you go through this list. Sometimes the relationship is one-to-one, it's really simple. Cells that divide when they're not supposed to, that is cancer, simple as that. Sometimes it's more complicated. Heart disease that kills people in the elderly. There's lots of different types of heart disease. Some of them caused by molecular changes to the matrix that holds the organ together. That's arteriosclerosis. Sometimes because of fatty deposits in the artery wall, I'll come back in a moment, that's molecular garbage inside the cell. Sometimes because of protein deposits in the spaces between cells. And sometimes because cells are dying. Sometimes it's more complicated than that. You've got diseases which have many components. So Alzheimer's exhibits molecular garbage called plaques outside cells, molecular garbage inside cells called tangles, and also cell deaths. Ultimately, how to fix this comes down to 4 different things, all of which begin with RE. You replace things that have gone missing, like cells that have died and not been automatically replaced by the division of other cells. You remove things that we don't want, damage that's accumulated because as byproducts of normal metabolism. Sometimes you need to repair stuff that you don't want to actually remove or replace, but you want to put it back into the same structure that it had before. And sometimes you preemptively reinforce the body in some way in order to make the damage that accumulates not actually translate into pathology at all. If we look at these 7 categories that I've written down, which of those 4 things they actually amount to, I don't have time to go into much detail, and we've got these specific therapeutic avenues that either are already being pursued so effectively within the biomedical industry that we don't even have to, or else we are pursuing them ourselves in our labs in California, or else in labs that we're funding around the world in various universities. The one that I'm going to spend the last minute on is the one that we're doing at Rice University in Texas. We're essentially addressing cardiovascular disease by getting rid of the toxin that accumulates in the artery wall and turns perfectly self-respecting white blood cells into things called foam cells like this, which are essentially undead white blood cells that are the beginnings of an atherosclerotic plaque. We found bacteria that can break down this toxin. We found out how they did it, what enzymes they were using. We eventually managed to modify those enzymes so that they could be targeted to the correct part of a human cell, and then we put it into human cells, and we actually succeeded in demonstrating protection of those cells from this toxin. Only in cell culture so far, but of course we're going further. We're doing a great deal more than this, where as you see here, we're doing something like 15 or 16 different projects now, both intramurally and extramurally, as well as having an educational initiative. This is really solid science. This is our research advisory board, 24 eyes acting out of the most stellar scientists you can imagine. This is very respectable stuff. These people would not be supporting stuff that was snake oil. We just had a conference last week in which our newest research advisory board member, the extremely famous George Church, gave a keynote talk. That was just last week. If you want to know more about us, then you can read our annual report or our research report, which are on our website, SENS.org, and of course you can also get involved. I'm afraid I'm going to have to stop there, but I will also refer you to this book that I published a few years ago, and which can tell you far more than I can tell you even if they'd given me an hour. Thank you very much.
Lots and lots of food for thought there, which I hope we will come back to very soon. Our next speaker is Hagan Bailey. He's Professor of Chemical Biology at Oxford University, and his major interests are incredibly arcane. They are all about the development of engineered pores for stochastic sensing, studying covalent chemistry at the level of the single molecule, ultra-rapid DNA sequencing, and synthetic biology of minimal tissues. I'm sure he'll explain all to you, asking nicely. But right now, he's going to actually talk at a far more conceptual, actually much more basic level. His talk is indeed called "Is Time to Get Back to Basics." So, Professor.
Well, it's a very different audience. I guess most of you are actually users of science, and I wanted to talk today a little bit about underlying basic science, and that we should be defending basic science. Now, at the institution I come from, Oxford, basic science has not always been in favour, as you can see here. This is Benjamin George, who was the master of Birkbeck College. But the UK has a fantastic record of research in basic science, and of course a lot of this has been translated into industry. And if we just look at medicine, we can see, we can go back to the invention of antibiotics, fantastic breakthroughs in structural biology, beginning with Dorothy Hodgkin, more recently with ribosome, DNA structure, DNA sequencing, mechanism of different drugs, different scanners, CAT scanners, MRI scanners, transgenic animals, stem cells, cell cycles, just an amazing track record that we have. But there are really problems, from my view at least, for the funding of basic science. And the research councils and other entities that fund science have landed us with a lot of, a great sort of burden recently, including strategic plans, delivery statements, impact plans, standards, believe it or not, involvement with stakeholders and so on. And I think they're really beginning to forget that really the best science, and of course that is what leads to the most significant applications at the end, just simply arises from funding the best people and just leaving them alone. And many of the people who have taken science forward in form companies have emphasized that their work really relies on basic science. And I just listed a few of the statements here from James Dyson, Chris Kent of GSK, and Graham Richards, who's an at Oxford. And the same issue has really come up in Big Pharma. As you know, Big Pharma has not really produced as much as you might think it would have done recently, given its massive budget. And this article by Pedro Patricasas, and he talks about management by objectives, autocratic organizations, strategic planning, scheduling, reporting, and all the stuff that we have to do at universities. So I've been fortunate to form one company in 2005, and I just want to tell you very briefly how that came about. I like this quote. It's actually one of the nicest things that's been written about me. Bailey speaks in the laconic baritone of a late-night radio DJ. If he has an entrepreneurial streak, it's not obvious. Well, I definitely traded my job to be a late-night radio DJ, but that didn't quite work out. But anyway, Oxford Nanopore now has 145 employees, and it's raised over £100 million in funding. And it was formed also with Gordon Singera and Spike Wilcox were co-founders early on. And what this company does is use protein pores. These pores are very tiny. They have 1/50,000th of the diameter of human hair, and we use those as sensors. And probably the best known application is so-called nanopore sequencing, where we can pull a piece of DNA through the pore and read off the bases as it goes through. And that has the potential to put the $1,000 genome and indeed a 10-minute genome into actuality. And the company announced actually last year that they could perform sequencing with this very small handheld device and do very long sequencing runs for those of you who know about DNA 100 kb plus sequencing runs. And this technology, and the technology that's gone before it, has obviously had a huge impact in a huge number of areas. But the main point I'd like to make today is that this really began as a basic science project. The question we were asking was a very simple question: how can a water-soluble protein assemble into biological membranes? This is how our pore-forming protein starts out. It assembles into a pore. And it's those pores that have been used to sequence DNA. But that's not the problem that we started out with. More recently, we've been trying to build synthetic tissues, and we've been using 3D printing to print aqueous droplets of mimic cells. So this is 3D printing the micrometer resolution. And we hope to use those to either augment tissues or to actually to be able to form hybrids with tissues in various ways. So we're forming another company called Oxford Synthetic Biology at the moment to exploit this technology. And again, this began as a completely basic science project. The idea was just to bring two of these droplets together, and we could insert proteins into the membrane between them and record currents through these proteins. So we could do so-called single-channel recording on a very, very minute scale, which has advantages in the lab, but it had nothing to do with building tissues when we started out. I think another thing of interest to you, I think, is the environment at Oxford. Many companies have started at Oxford, of course, other universities as well, and has influenced several students in my lab to start companies. I'm probably going to run out of time. I'm getting about to get a few minutes. No. So several companies in my lab, several students in my lab have started companies. Most of these are in biotechnology. One is actually Maria's called a cosmetics company. And also, I think in Oxford we've also gained from our interactions with the students. Dan Perez, who should be here today, but I think he's he's too busy, who founded Marbler and Oxbridge Biotechnology Roundtable. So we've, as academics, really gained from his energetic efforts in entrepreneurship. again, my point is that none of this required any planning by funders. None of this required any impact statements and all the horrifying things that we have to write. It just happened through the right environment. And one of the, I think, the most insidious things that we have now in the UK, and it certainly applies in other countries as well, is so-called priority area research. And my field is called chemical biology. And I think as a result of this priority area research, we now tend to be followers rather than leaders, because the research councils tell us in a very broad way what to do. And science in the UK, I think, was a lot better when the scientists themselves introduced new ideas and led fields. So I'll just move on a little bit more quickly now. And interestingly, I think this way of doing science is actually not the policy of the government. So if you read George Osborne's speech from November 2012, it's actually a very good speech. And he argues against top-down science. And in fact, the simplest thing to remember from his speech is that innovation is not a sausage machine. But a few other things that I think we need to deal with, but I won't spend time on today, but obviously of interest to you. I think a lot of research has been very secretive, and there's a big move towards open access research. One outfit that's doing a really good job in that area is the Structural Genomics Institute in Oxford that does so-called pre-competitive research with a large number of drug companies, and they share the data that comes out of that. I think another issue, I think in the UK, I was in the States for a long time, but returning to the UK, is that we need a better deal for young academics. I think in particular, the PhD degree here has become more a training degree rather than a research degree, and people at the assistant professor here level here get a rather poor start, and particularly in my field, the multidisciplinary programs are really quite frowned upon, and I think all of this has led to a dampening of the research ambition. So, in summary, I think we need to support basic research. You guys, all of this underlies what you do, and we need a really unfettered approach to that. The funders should, as I always say, provide the theatres, but not write the plays that we're going to put on. We need to increase the basic science budget. I think the country can well afford to do that. And we need to expedite the transfer of knowledge and give a better deal for academics starting up. And the good news is, this is from 2009, that this doesn't actually, in the big picture, cost a lot of money. So this is a slide showing the research expenditures of the leading pharma companies in the world. It's GSK here and AstraZeneca. Actually, the UK budget deficit at the time was rather large. You had to add up all these research budgets to get the UK budget deficit, which is a little bit slim. But the entire Research Council budget of the UK lies here. It's much smaller than many of the drug companies. And the research budget of the University of Oxford would be this large, and that includes our efforts in ancient Greek. So, thank you very much.
Thank you very much, Bailey. Again, lots of food for thought there about basic science and how it should remain unfettered. Now, we've got somebody here who's going to be taking things in altogether other direction, and that is Giuseppe Battaglia, and he's Professor of Molecular Bionics at UCL's Department of Chemistry, and he's also affiliated with the MRC's Centre for Medical Molecular Biology. His specialism, bionics, involves basically applying biological methods and systems that are found in nature to emerging technologies, in his case, nanotechnology. as he can be explained in his book, which is called Bionics at the Nanoscale. So let's hear it for Giuseppe Battaglia.
I'm also have to watch the slide. I'm going to speak really fast. However, I have a different accent and not so you're not going to understand anything what I'm saying. But I'm actually going to try to make a much calmer presentation and prefer to skip the slide if it's necessary. So please give me a try on the time. So let me start with actually do a disclaimer. And you know, our job in academia is actually expansive science, but most of the time actually what we do is managing people, and actually most of the work is done in my lab, without being done by me. And so I like to start out with our knowledge of the people that have been working with me under my supervision or with me as collaborators, and of course the different agencies they actually provided their funding for us to work and produce some work. Okay, let's start with the semantic exercise. What is bionics? In medicine, bionics is very much the technology or the science of replacing damaged parts of our body. So you can have a bionic eye, you can have a bionic heart, a limb, an arm, etc. And this is already a well-established technology. It's very multi-billion-dollar industry already. There are several products there which are, of course, sold in the hospitals, and they are also funded in the hospitals doing bionics in that sense. But of course, I'm not going to sell that to you, because probably for you bionics is more sci-fi. And probably every time I mention the name bionic, you have to remember the bionic man and the bionic woman. And if you put that in Google, you come up with a lot of nicer pictures than actually the artificial art and everything. And I particularly like this, where you have the bionic woman and bionic man. And this is a comic magazine, and in most of the front cover, it's always a bionic woman leaning against a bionic man. So it says a lot about what to do with that. But the sci-fi, also taking inspiration from the alien stuff before, inspires science in many ways. And of course, this is also goes into philosophy. How we can apply the same technology not just to replace parts, but also to improve certain parts. And I'd like to bring this to a different level, going down to the molecular levels, and explain why I have a molecular bionics, not just bionics. So my take on nature of biology is very reductionist, as I am a scientist, and we like to simplify before we complicate. And so if I plot what is an organism like this embryonic human being in there into an axis where I have the size in here and the number of complexities, or the number of species you like. This is more or less what it looks like. And nature is beautiful because it allows our diversity and our complexity through an hierarchical arrangement of molecules, macromolecules, supramolecular structure, and everything. And everything goes down to a really nice level of complexity to a bunch of molecules. They are nicely modified at the chemical level, but most important, they are then put together into a very, very precise way. This concept in nature is known as a self-assembly. And then most importantly, the way the nature controls that is by generating compartments, sort of environments where you can actually control such a sophisticated process. So for many years in our group, we've been trying to exploit the same concept. Of course, keeping an eye on what is the translation of this approach. And of course, trying to address technological challenges, mostly coming from the clinics. And incidentally, I'm originally from Sicily, so we like to work on drug delivery. And of course, diagnostic is another big chunk of things where you can actually intervene in many ways, so transforming the nature into actually studying nature. And then, of course, the recent event bringing forward also the synthetic biology approach, where you can actually redesign nature into many components, and maybe transforming biotechnology into an in vitro one, so without the needs of animals or other living systems. So let me focus, of course, in drug delivery, my favorite topic. And the concept is not really easy. It actually goes down to as long as necessary drugs have been designed. Not only you need to design the chemicals or the biologicals nowadays, you also need to formulate them and then make sure that they actually go to the right site in the body. And this concept goes down to Paul Ehrlich, one of the Nobel Prizes actually contributed to discover antibody, and he came up with a magic balance concept, how to actually home in our devices or our drugs into that. And that is not easy exercise, because you need to go through a series of biological barriers into the human body, and these are not trivial. because we still don't know most of them, because biology is still studying them. And so it's a very important exercise to combine all this knowledge and then transform it into engineering solution. So, but because we're lazy in our lab, we do not want to invent new stuff, so we again copy nature in our actually nature found solution. Because nature does not just set up barriers, also finds ways to actually make possible that you can go through these barriers. And as a consequence of that, there are many biological structures in our body that actually move across these barriers, like the red blood cell, immune cells, and not need to explain what a stem cell does. And then, of course, other more complex and smaller systems like microvesicle, like proteins and certain proteins. And then there are the world of pathogens. Viruses and bacteria continuously trying to enter our body, and they are very sophisticated way to actually deal with those biological barriers. In some cases, in particular like the viruses, they can go all the way up to the final frontiers, which is at the inside of the cell, which is basically the building block, the atom block. So, if we, for example, take inspiration from these natural events, natural system, then we can adapt physics and chemistry in copying some of these functionalities in artificial surrogates. And of course, this is not just an economic exercise, because the synthetic material costs much less than the biological one. It's also an exercise in terms of stability, because you can control exactly what you put in your top, because you know exactly what chemical modification you can do. And this is exactly what we do in our lab massively around many different nanostructures. I'm showing you a few examples of pictures taken from my research, where we try to copy some of the structural functionality of viruses. And of course, then you came up with a platform technology that allows you, in many ways, to encapsulate things and deliver them. But how can you go from A to B in a human body is a major question. And there is also a research activity, both fundamental and applied, trying to tackle those biological barriers again. And of course, you need to go one by one. First of all, many people don't know that actually we are made of water, probably we do. But what we don't know is that this water is not just free water, it's full and rich on many proteins and other molecules. What I put here is a series of proteins which I downloaded from a protein bank. And then you see that basically everything filled up the space of water that you actually encounter in the blood. So it's a very crowded environment. And of course, designing something that can actually operate in that is very critical. So you need to find a way to then go through all of these, etc., and avoiding the perturbation of this environment to the negative way, and favoring the perturbation of the environment to your own benefit. So I'm going to do this, because I have only 2 minutes, and skip ahead in time with just this. So you can do all of this, and eventually find out a beautiful way to engineer a solution. What I'm showing you is some data we recently finished after 4 years of work, where we try to adapt this approach in getting inside one of the most difficult organs in our body, which is the brain. The brain is highly guarded and it's very difficult to assess. However, again, nature has its own way to regulate this access. So we developed a technology that can target those process called transcytosis, and eventually our best formulation can get 80% of what the overall injected materials into the brain. 80%. And this is one of the, actually, if you compare to the market, it's actually not bad. I think the best is about 10%. 8%, so only of the materials that you inject is actually ending up into the brain. So why is that? Let's pose a question. If you want to actually selectively target a specific part of the body, but if you want to really do the homing exercise, you need to have some chemical affinity, because they're chemicals. So you need to find other chemicals that stick to that particular one. And the king of this is the antibody. The antibody technology has allowed us to do this sort of homing exercise by designing proteins that can stick to other proteins and non-proteins materials as well. And it's beautiful. They're very affine and they're very nicely selectively. However, this type of interaction, the control, that binding, the locking exercise, happens in a range of about 30 to 40 nanometers. So it means, in order to actually have my affinity, I need to have my antibody there at 40 nanometers thick. And then, of course, this is the problem. If I inject my drug in the body, the drug will go everywhere. And will only bind to the points where it should be binded, but inevitably it will bind to other parts, and that's why you have side effects. how does nature address this problem? And this is beautiful because actually the nature does this by a process called taxis. So it moves its all machineries according to specific chemical gradient or other gradients. Chemotaxis in particular show like this immunocell following these bacteria. Does this very effectively. So we take inspiration from this and try to adapt this concept of chemotaxis into our nanoscopic devices. And then eventually, let me show you the final slide, because otherwise this girl is going to kill me. Find something to throw at people. And then basically, without going into much details, the long story short to show in showing this picture, basically we can actually devise our system, and not only we can actually have this ability to interact with biological period, but also the ability to follow small chemical gradients and therefore targeting things over really long distances. This is a petri dish where we have our system, which is equipped with this molecular gene, and then it can actually quickly diffuse towards the source of the chemical structure. And I finished with that, and hopefully I haven't talking too fast, because really I know my accent can be really tricky to understand. Thank you very much.
Going from the wildly conceptual, I was saying, the basic and also quite novel approaches, we're going to something really quite practical now, or at least it sounds like that. Our final speaker is Mohammed Haji Hosseini, I hope I've got that right. He is a senior lecturer in development and neurobiology at the University of East Anglia. He's very interested in trying to do something about the increasing prevalence of eating disorders and the strain it's putting on the global economy and the global health budget right now. He's got a novel weapon, he thinks, up his sleeve, and he's going to tell us all about it in his talk. Stem cells, a novel weapon against eating disorders. Let's welcome Mohammed Haji Hosseini.
I'd like to thank the organizers for inviting me to share some exciting results from my lab with you. This is novel and it's up and coming. And this revolves around the population of novel population stem cells that we've recently discovered in the adult mouse brain, and we think we could harness the power of these stem cells to treat eating disorders. This banknote that you see up here is going to hit the high street in about 4 years from now, and on the back of it is Jane Austen. What the legal views are on which on the back of banknotes. I think this statement down here doesn't really concur with what's happening in our society right now. Because with the increasing amount of high-calorie foods that we're consuming at the wrong time and the wrong amount, I suggest that the poll of people would suggest that there's no other enjoyment other than eating rather than reading right now, particularly children. And that's translating into a massive problem worldwide, not just here in the UK, and that's the problem of obesity. So you have a measure of calculating overweightness and obesity, and that's body mass index. It's a certain formula that I'm sure for undergrads. It's a crude formula, but it roughly categorizes you into overweight, obese, and morbidly obese people. What is alarming is that when you think that a lot of our traits have taken millions of years to develop, this trait, on a data taken from the Center for Disease Control from America, shows that in a 10-year period, every state in the US has moved up the overweight and obesity and morbidly obese scale significantly. And this is 2009 figures, so we're actually way over that now. This is not a US problem, it's a worldwide problem. In 2010, roughly 26% of adults are considered overweight or obese. 30% of children, that's very alarming. And then by the time we get to 2030, the current rate, roughly half the population is going to be affected. At the other end of the scale, you have adolescent-related anorexia, mainly in women, but more recently, something called manorexia, this has now been documented in Bronx, when young men are trying to look lean and fit, and they're actually starving themselves to death. But more importantly, with an increasing aging population, if you have grandparents in their 70s and 80s, like I did until recently, you will see that actually they skip meals. There's an anorexia develops in these elderly people, and that's a genuine problem. And that anorexia exacerbates the problem of aging because of malnutrition. So what are we going to do about this? Well, the problem with obesity is twofold, really. It puts a burden on the individual because of need for wider toilet seats and airline seats and so on, but it also puts a massive burden on health budgets. And the main problems highlighted are here: diabetes, cardiovascular problem, arthritis, the sheer weight of these bags on the joints of the knee. And more interestingly, recently it's been discovered that fat cells start secreting things acting like an organ that induce cancer, certain types of cancers. More alarmingly, it seems that pregnant mothers, when they consume these high-fat diets, it has an impact on fetuses. So we yet have to see the effect of that on the subsequent generations. And in terms of numbers, basically in 2007, £4.2 billion have been spent on tackling these problems, and this is projected to rise by 2050 to 9.7 billion. So we're talking about big money. What are we going to do and what have we done about it? Well, dieting is not a solution because it was in the word by now. And in fact, yo-yo dieting is actually worse than dieting. It seems that once you go through a certain barrier of weight loss, there's a physiological resistance to further weight loss. So that's why it's not working. So people go back to exercise is not an option. I have a table here. I was going to read out how much it takes to burn a Mars bar and so on. And we don't have the time to exercise. We don't have sedentary lifestyle and busy lifestyle. Gastric banning is proving effective. It's not a solution for everyone, and it has side effects. And drugs have been developed, but some of the side effects are really massive and not tolerable by everyone. So those are not the options. We could try public education and policy making changes, introducing taxes on high-fat food as they've done in Scandinavia and other places, but that's for the long term. The solution seems to be in the brain, and experts agree that because the brain controls appetite regulation, eventually the solution is going to be in the brain for a sustained solution. So our question is, can we rewire the brain? So how is appetite controlled? I'm going to show you first if this works, a 3D a rotation of mouse brain, and the area that regulates appetite is down here called the hypothalamus. So if you take a section through the mouse brain, that's what you see, and a coronal section will show you that this region, tiny region of the brain compared to other sizes, this is the area that regulates appetite. Okay? It's composed of clusters of neurons, which I'm named down here. I just want you to remember arc. Down here there's a hole called the ventricle, and down here there's a structure called median eminence, and this hole here is called the third ventricle. So that's roughly to orientate you of where you are. Now the way appetite is controlled is, within the arc there are two clusters of cells, two types of cells. The red ones suppress appetite, and that's the default pathway. So this is the skip the animation. So this is what happens by default. These neurons fire and they suppress appetite. To make you eat, you have to make the conscious decisions in response to certain signals, and that happens as a result of the inhibition of these pathway by these green cells. Okay, so the balance of activity between these two neurons determines whether you're hungry, you should eat, or you're full, and you shouldn't. And that happens in response to certain signals which come through the blood, stomach, pancreas, and so on. And one of these key ones is secreted by fat cells called leptin. We know this is a signal because if you knock out the gene for leptin in mice, the litter mates go obese within weeks, and the ones that are carrying the leptin gene and its receptor, they're fine. Okay, so there's a lot of data to support that this is what's happening. Can we do, how are we going to modulate the appetite regulating circuitry? Well, a ground rule for brain function is that brain function relies on three things: for the cells to be in the correct numbers, in the correct position, and the correct connectivity, as I showed you in the diagram. You have to have certain numbers of cells, and we think by estimate we should be about 4,000 to 6,000 of these. And we know numbers make a difference because if you transplant cells from the brain of the wild-type mouse into the leptin division, you rescue the obesity. What that also tells you is that this environment is receptive to receiving new cells. Okay. So are there pathways that we can, how can we modulate these numbers? And this is where we come in. Recently we discovered a population of cells called tanycytes, and these are found in the bottom region here. They look like neural stem cells during the embryonic period. They have this radial appearance. And we had to convince lots of our colleagues that these are stem cells, and needless to say that these cells are very close to the appetite-regulating circuitry. The first evidence was that these cells express markers of, well-characterized markers of stem cells, which are listed on the left. I don't have time to go through them individually. The second piece of evidence was that these cells actually divide, and nobody had shown this before conclusively. So what we did was we injected mice with a substance called BRBU. This is taken up by the DNA of the cells as they divide, and you can detect this substance later. So the sections down here show you pairs of dividing cells in mice that have been taken up BRBU through drinking water and analyzed at subsequent points. So these cells down here are dividing, just down there. Next, the more challenging thing was to show that these cells are actually doing something. And for this, essentially what we needed to do is, this animation is very slow. Okay, I'm doing animation. Essentially what you need to do is to label one of these cells and follow their fate. If they generate more cells, they're characterized as progenitor cells or cell front. And to do that, we used a very genetically modified mouse in which if you inject tamoxifen, you activate a label means that one of these cells or small clusters of these cells. Then if you sacrifice these mice at that time point, the cohorts of these mice, what you find is initially you label that population or a few of them. If you wait a little bit longer, this population amplifies, as you would expect, and now you start seeing new neurons or cells appearing in the adjacent areas, nuclei that regulate appetite. And the animation is completely messed up. And then if you wait a bit longer, these cells extend axons, and these are well-characterized appetite-regulating neurons. So basically, how do we take this to bench? What we're going to do is to work on these tenocytes and either produce more appetite-suppressing neurons or reduce the appetite-promoting neurons as a long-term solution. And what are the challenges en route? Well, one of these is we need to know what types of neurons are produced by these, are these appetite-suppressing or promoting. We need to have a better understanding of the circuits themselves. What is the upstream regulators and more of the downstream regulators? We need to work with mathematicians to work out actually, are these cell numbers critical? What is the minimal critical mass of these cell numbers, and would a 20% increase or decrease affect the circuitry? We need to identify the genes that regulate the division of the behavior of these cells, and we already got some handle on that. And then, armed with this knowledge, we will design and deliver drugs into this ventricle here, which is close to the cells that we've identified, and that will modulate their activity. My last two slides are something beyond appetite regulation. The question that might go through your mind is, well, what are these cells normally doing anyway? We think this is what's happening. The division of these cells we found actually progressively slows down as the mice age. So we think what is happening is that these cells are constantly supplying the circuits with new neurons as the mouse is going on. And neurobiologists will tell you that this sort of phenomenon will give a kind of plasticity to function of the circuits during that period. Okay? And this might be susceptible to food we eat and so on, genetic factors and so on. And as we lose this plasticity, then the adult, the circuits that regulate appetite might become fixed. Beyond that, because we've identified a new population of stem cells in the brain, we're very excited because you could use these for other purposes. And one of them, we think, is we can direct these to form oligodendrocytes to replenish MS, or new neurons to treat neurodegenerative diseases. This work has been the result of some fruitful collaboration between some people in my lab and Germany, France, and Finland. And I just want to acknowledge the work of Tim Goodman, very hard-working postdoc in my lab, and of course the funders who funded this work. Thank you for your attention.
I think what I'm going to try and do now is take some questions, and I'm very much hoping that some of you will have lots of good questions, because we've had an awful lot of good stuff, a lot of approaches, novel approaches, very specific approach. And I think the question is, how are we going to make this actually translate into real world stuff, is what people really care about. So perhaps you could just answer that question and then we'll take your questions from the floor. transported.
How are we going to translate this specific real world stuff? Well, we're already doing it. One of the number one ways in which SENS Research Foundation chooses what to work on is, since we're quite small, we choose to work on the things that give the best bang for the buck, and that usually means the early stage stuff. But luckily, that means that we can exclude some things that are already in clinical trials. There are a number of different stem cell therapies, for example, including for age-related conditions, which are moving in that direction, and that's a very intrinsic part of what we're going to do.
As always, if we're already doing that, I think we need more funding to do it because funding is really the main driver of national limitation.
We also need to train a new class of scientists that can do this translation exercise. Because of course it's all about exercise, all about translation, but the lag, the time lag can be very long sometimes. And you might lose a certain expertise and skills by jumping into one of the others. So maybe also generating new T people, so people that can actually sit between the two of you and try to understand fundamental science and bring it into a real world application and might actually be the way to go for it.
That sounds like kind of natural thinking that you were hinting at, I think, really.
Yeah, I guess from my viewpoint, I see many scientists in New York who spend our days doing science, but really new things, really effective things, tend to suddenly come out of nowhere. The penicillins, the DNA sequencing, the scans, and they have an almost sort of exponential rise from the baseline. And when these things do appear, people like you guys, entrepreneurs are around, and other people who want to exploit these technologies and take them to the marketplace. So I really do believe that we need to maintain that underlying basic science to allow these really revolutionary change our lives to emerge.
I see somebody usually down here.
I think perhaps the opposite was the point of Hagen's talk, which was that it's not about what it can be applied to, it's what this guy's research can do in its own merit. I was wondering what are some things that you'd be excited if you had a special golden content to put anywhere, what would you want to fund?
Well, I would we have, say in the UK, a very limited science budget. It sounds huge, but maybe 3 billion or so, depending how you look at it. And really, I wouldn't want to do what you say. I wouldn't want to be like research councils and do surveys and sandpits and whatever, and come up with things that we ought to spend our research on. I would like to make sure that we maintain the best biologists, chemists, and physicists, mathematicians, and let them come up with their own ideas. I think that's where we really need to start. So I'm not a real believer of this sort of incremental, slow struggle towards a goal. I think that we need to maintain people working in a certain area, just as you would have artists painting paintings, and then suddenly, almost like magic, something new will appear.
Yeah,
but we need to be in a position to exploit that when it happens.
I think it takes all sorts. I think there is a very significant difference, again, probably getting at this a little bit, a very significant difference in the mindset that exemplifies the best scientists, the best basic scientists, discoverers of how nature works, and the mindset that exemplifies the best technologists who exploit knowledge about how nature works. I think sometimes what is missing is the bridge between those two, not necessarily people who have that disciplinary skill, though that would be great, the more of them will throw out better. But simply the process, having a system for more powerfully identifying when a body of knowledge has reached a point of technological exploitation that the people who of technology may not actually be the best people to employ. You know, I think that system is missing not just in the UK, but throughout the world.
Can I see what I can do?
So, aging is a big problem. And one thing that all of you did mention is that funding is a problem for all of your disciplines. Now, I know that AUBREY mentioned that if you look at the symptomatic sort of phenotypic causes of aging, it's looking at sort of tackling there at the phenotypical level. Whereas on the other hand, aging, you want to tackle things from the very basics, which was what your talk was about. And Berkeley said, well, let's do something completely different, so let's modify the way that they work. So how do you think that those three can connect, and how much money or effort should be put into each of the three disciplines in your opinion?
Good pressure there,
Maybe that Morgan says might answer the question. I'm going to support Hagen's statement in that we need to fund science across a broad spectrum of disciplines. The reason is the work I described came out of research into bone disease, completely unrelated to brain or appetite. It just happens to be that the same gene that regulates these cells also regulates bone development. And suddenly through researchers and collaborations and our knowledge from other fields, we've been able to translate that and take that into completely new research.
Yes, and I mean, actually, the example I gave of the research on how we are tackling cardiovascular disease also exemplifies this. Here we have actually technology which was originally invented by people who work on environmental decontamination. They're not working on anything biomedical at all. And, you know, just suddenly it became apparent, so a few of us, that this could be used for a completely new application. So this really kind of underlines how it's really impossible to judge what basic science ought to be funded most heavily and least heavily. You've just got to rely on curiosity of best people. However, it also does mean that we need to somehow find better ways for people to notice the applicability of a certain body of knowledge to a certain problem and not having just be forgotten.
And sadly, I'm afraid this is where we have to leave it. I think the one thing I was just cautionary note, the one thing you probably all would be important to hear is a story from back in the late 80s when I was a computer journalist and IBM researchers in this lovely ivory tower where all the IBM researchers do their thing. And the poor bastards used to phone us up and say, no one was listening to them. So people phone us up as journalists just to beg to tell us their stories. Tragic. But anyway, I'd like to thank you very much our speakers today, AUBREY, Big Bailey, Giuseppe Battaglia, and our Higher Education Studies. Thank you.